Hello, everyone, and welcome to today's webinar, Predicting, Tracking, and Controlling Diseases and Epidemics to Prevent New Epidemics and Pandemics that can be amplified by climate change. I'm Susie Valdez of LabRoots, and I will be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Abbott. To learn more, visit them at abbott.com. Now, this webinar is educational and offers free continuing education credits. Please click on the continuing education window at the bottom of your screen to obtain those credits after the webinar. And we encourage you to participate today by submitting any questions you might have during this presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. You may also submit any technical issues here as well if you have any trouble seeing or hearing this presentation. I'd now like to welcome our speakers, Dr. Mary Rodgers, Principal Research Scientist at Abbott, and Professor Tulio de Oliveira, Director at the Center for Epidemic Response and Innovation in South Africa. Dr. Rodgers and Professor de Oliveira, welcome to both of you. Dr. Rodgers, you may now begin your presentation.
All right, thank you so much for the introduction. It's a pleasure to be with everyone today, and I'm looking forward to telling you about the work that Tulio and I are doing as part of the coalition. So with that, I'll get started. So I'd like to start by giving some context for first, we'll start with disclosures, actually. So I want to make note that support for this program is provided by Abbott. We're both presenting at the request of Abbott. And the information contained in this presentation is from the speakers. It does not represent new or implied product or regulatory claims of the products by Abbott. So to give you a little bit of context for the work we're going to be talking about today, I want to share that we are looking at how viruses mutate and how that may impact the detection of viruses as we have diagnostic tests developed against them, whether those are serology tests or molecular tests. So in the case of serology tests, if the virus has mutated, that could impact our ability to detect either the antigen or the antibody. And of course, if there's mutations that are in the target region of a molecular test, those could be impacted as well as a virus is mutating. In fact, if we look at the viral diversity of two of the viruses that we make diagnostic tests for, HIV and SARS-CoV-2, and we plot them on a phylogenetic tree, we can see there's a difference in the level of diversity between these two viruses. In fact, at the same scale, the SARS-CoV-2 phylogenetic tree is just the period at the end of a sentence, whereas in comparison, the HIV tree is much more diverse and has a lot of different sequences represented in it. And that's because the number of mutations that separate these different strains differ between the viruses. So if we want to make a test in a conserved region of SARS-CoV-2 that's less prone to mutation, that's a lot more straightforward than making a test that is targeting a region of HIV that could be the same in every strain. So knowing about the sequences helps us to design those tests to mitigate the impact of viral diversity. And we've been doing this work for decades. In fact, we began in 1994 when we started doing a viral surveillance program here at Abbott. And we've been primarily focused at the beginning of the program on HIV because it's so diverse. Since then, we've gone on to add new viruses, hepatitis B, C, and Delta, because those viruses also mutate frequently and can change and potentially impact tests. So over the course of the program, we've collected over 100,000 samples and published over 150 papers describing the work we've done in the program. We've also deposited thousands of sequences in public databases for all of the work we've done. In addition to the viruses that we've looked at here on this timeline, we've also started doing work in the public health area, where we've collaborated with countries doing serosurveys to be able to identify how prevalent these viruses are in their communities and have targeted services to engage and potentially treat or prevent new infections. So this includes partnering with the Georgian HCV elimination program, which we started doing in 2015. And then 16 and 17, we were working with the Democratic Republic of Congo to look at the prevalence of Hep B and C. And more recently, we've also started doing a hepatitis B elimination program in Pakistan, where we're working with our partners to understand the prevalence and identify patients who need treatment. So along the way, we've also discovered new viruses. And these are diverse strains of the viruses that we've been studying. So in 2011, we found the second known case of HIV group P, which is a very diverse strain. It was one of the farthest one out on that phylogenetic tree I showed. And to date, there's only 2 cases that have ever been identified. But what's important is that we were able to detect this with our tests before we knew it existed. And that speaks to how we've been keeping mindful of viral diversity as we design our tests at Abbott. Further down the timeline, we've also found rare strains in Cameroon and the Congo and characterized those over the years. But something that we saw in 2015 really stands out, and that's when we identified a new virus. In addition to the known viruses we're looking for, we're also continuously looking for new pathogens that might present a threat. And so in 2015, when we found a new virus, we developed prototype diagnostic tests to try and understand the potential impact of that virus. We didn't see any cause for alarm. It's not the next pandemic, but it taught us a lot about how we can move quickly to respond to new viruses that are emerging. And so collectively, all the work we've done in the program has led us to the fast response we had in 2020, where we developed over 7 different assays to SARS-CoV-2 in a matter of months. And so with this in mind, we've now continued our work to understand new pathogens as we launched the Abbott Pandemic Defense Coalition. So we're always under the risk for a potential new pandemic threat. And that is caused by a couple of different components shown here. The first is the risk of known pathogens, so things we already know about that continuously reemerge from time to time in outbreaks. But the second cause is the viruses and other pathogens that we don't know about yet. So those could be causing illnesses that never get diagnosed, and that accounts for a pretty significant number of cases in these disease states shown here. So if we knew what was causing those illnesses, we would be able to better define and meet the needs of these patients. But as they go undiagnosed, there's a potential that something could be circulating that we don't know about yet that could cause a new pandemic. And so that's where the coalition comes in to try and make a difference. So if you look at our map here, and you can see that we have 20 different partners on 5 continents that are doing the work in the program. And I'll show a little bit more about them in a little bit. But what makes this program unique is that we have sites that are very strong at being able to recognize cases that could potentially be a new pandemic. So there's access and geographical locations to patient samples where there might be a higher risk for new illnesses to emerge, whether that's because there's close proximity to reservoirs of animals or vectors where new viruses or other pathogens could cross over into the human population, or because of the geographical location where there's a lot of people coming together from different places in one location. And that always allows pathogens to spread as they come along with the people who are traveling. And all of these sites have excellent technical capabilities to be able to do the work of the program and identify new pathogens. And so the program works by first identifying what's in a sample. So we have clinicians around the world who are looking for cases where they've come in sick and the person has tested negative for usual pathogens that would cause that illness. So we are able to then do sequencing on those samples to identify if there's something new. And that takes a lot of sophisticated bioinformatics to be able to identify what's new in a sample. But once we find a new virus, and what really sets our program apart is that we can go on to the next step in the development phase, where we can develop research use tests that are molecular, serology, or rapid diagnostics that we can then deploy to all of our sites, especially the one where the first case had been seen, so that we can start to do initial research to understand how far something has spread in a surveillance study. In those same kinds of scenarios, we can learn a lot about a pathogen early on. And that information that we learn, because we can identify positive cases, is what we can use to help public health communities to take appropriate measures and make decisions that will protect people. So in the end, that's how we hope to see the coalition making a difference. And we're off to a good start. If you look at our track record so far, we have 36 publications, and you can find more information about those on the website. I won't elaborate them on this webinar, but I invite you to take a look. We also have more information about our program in some episodes that have aired online at abbott-discovery.com that show in depth how some of the partner sites are working. In particular, there's one that highlights the work that my colleague, Dr. Oliveira, will be sharing in the next part of this webinar. And then we've also been featured recently in Time Magazine and showcased the work we've done over the course of the program. So this is what our map looks like, and you can see where all of these sites are located as part of the coalition. We have 7 sites in Africa and 1 in South Africa that we'll be highlighting today is the work done by Dr. Oliveira and his team here at CERI. And so with that, I'd like to hand over the presentation to Dr. Oliveira. Welcome, Tulio.
Yes. Thank you very much. And it's a great pleasure to be here with Abbott today. As we speak from you, we just launched here in South Africa the biggest investment on biomedical research innovation opened this week. Is a state-of-the-art facility based in Cape Town that it really have some of the most advanced methods. Just a second, adjust new computer. So what we thought that would be great to also do here in this week, given that we are launching the largest genomics facility in Africa, to also with some collaborators highlight another program that we have been that we are launch, and that is a program that affects all of us. It's something that's going to be really happening all around the world. It's basically how climate change is shaping the future of epidemics. And a global initiative that we are launching called CLIMADE, that means Climate Amplified Diseases and Epidemics. This global initiative has many partners. We have over 100 scientists collaborating in dozens of locations across the globe. And that also involves collaboration with foundations such as the Rockefeller Foundation, but also with companies like Abbott to make sure that we can detect quick new epidemics as it emerges, especially caused by climate change. So just to start my presentation, I think that's quite clear. The world have really been abusing the environment. And I'm just going to show 3 photos of a very impressive photographer that show the effect of climate change in the world. So not only we are completely abusing fossil fuels, we are really polluting the earth. And that is causing major effects. It's causing effects on the environment, on climate change. We are losing big environment, both at forest, at the Arctic, at Antarctic, and that creates a problem to humanity. We start in being much closer contacts with animals that we would be from different efforts. And what it's caused, as we degrade the environment, we increase also the extreme events, and I don't have to say how many extreme events we had recently of floods, of big fires, of storms, of droughts that begin to fuel large epidemics. Not fueling epidemics, but also fueling diseases and fueling sometimes this in a direct or indirect way. So for example, one thing that's also fueling that is what we call climate migrants. It's when some people that are affected by a climate calamity, or sometimes a simple thing as losing a harvest, they have to migrate to towns, and they go live in very poor condition, which really can also help the spread of diseases. Yeah. But one thing that we have done in South Africa and with partners around the world, we really learn how to respond to epidemics. We learn how to do science in real time, and also having access to sometimes the best technology, where people do not even expect. If you walk today in our building that we launched this week, it is really of the level of the top laboratories in the world, and having access to very automated diagnostics so we can respond in real time. One good example was the role that South Africa and the center that I direct, CERI, had on identification of many variants, and the most extreme variants such as Omicron, and how we could do that in hours, not in days or weeks, really highlighting the previous experience of dealing with epidemics, when coupled with high technology, can really help to respond to epidemics in real time. And what we are talking here, unfortunately, is not a small problem. A very important review was published in Nature Climate Change, middle end of last year, highlighting that around 60% of all pathogens in the world will be amplified by climate change. And here we are talking many different kinds of pathogens. Just to ground you in this graphic on the extreme left, I have the climate events, such as warming, precipitation, floods, drought, storm. On the middle of the graph, I have the way that pathogens are transmitted, for example, vector-borne or waterborne. And on the right, I have the specific pathogen. So one thing that we know is that viruses is going to be getting more normal, and they're going to be increasing transmission by vector-borne, such as mosquitoes, such as ticks, but they also going to be increasing transmission by waterborne and airborne as we just go to a pandemic. So if you get the details about viruses, we are talking about, just getting a closer detail on that graphic, we are talking, if you just get a look on virus transmitted by mosquitoes, that's going to be amplified, we have on the middle the species of mosquitoes, that's the ones that we worry. For example, one of the ones that we are very worried is the subspecies of Culex. Culex is a subspecies of mosquito that live at cooler temperature and high altitude, which can spread these diseases to many areas of the world. And that we are talking about epidemics that had happened multiple times in many countries in the world, which we have long experience on that. So we are talking about, example, chikungunya. As we speak today, we released some results with colleagues on Brazil on the most extreme chikungunya outbreak in Paraguay. So we are talking about over 100,000 cases. We send a team to go help the Paraguayan government to sequence and characterize that. And that not only 100,000 cases, that was caused by a 2 degrees increase in the temperature, followed by high precipitation, mosquitoes, and the highest outbreak ever in Latin America, with multiple deaths that have never seen of this virus. But chikungunya do not only affect Latin America, recent epidemics in Italy and also entering North America. We are talking about dengue fever that's spreading. We are talking about another virus, Rift Valley fever, that has massive pandemic potential. Last year, we managed to work with colleagues in Kenya to characterize and control an outbreak of Rift Valley. West Nile is completely increasing the spread around the world. In the US, you are having big outbreaks, West Nile, and then we talk about yellow fever, Zika, or a parasite like malaria. Unfortunately, they are all being amplified and it's going to be more common. And what we need to make sure is that they do not evolve to be more pathogenic. On water, but we are talking about a lot of affection of drinking water. I don't have to tell what a problem that is. In Durban, recently we have a flood with a massive outbreak of E. coli. As we speak, following floods in Malawi, we have the biggest cholera outbreak in the African continent. And we are talking about cholera, E. coli, leptospirosis, but we're also talking about viruses such as hepatitis virus that can infect the water stream and cause outbreaks. Yeah. So that's the kind of pathogens that we are fighting. Yeah. And the way that we do that, it is by creating a global consortium to generate the knowledge, to develop the tools and interventions that we can not only track, but that we can predict and control diseases and epidemics that amplify the climate change. And that's the name of the consortium, CLIMADE. It means Climate Amplified Disease and Epidemics. But we want to go away, step forward, not only to predict, track, and control, but really use that advance to prevent the spread of global epidemics and pandemics. If we can detect these pathogens, characterize, have diagnostic and therapeutics before they spread further, we can control outbreaks to become epidemics and epidemics to become pandemics. So where that should be done? Yeah, it should be done especially in the Global South. Why the Global South? Because the Global South is the region of the world that are experimenting more severe climate change events, especially extreme, and have a long experience to deal with these pathogens. I hope we have shown the world how our experience to dealing with epidemics could really help the identification of variants of control of COVID. Yeah. But this is not just a Global South program. It's really a global contract, a program that brings together a consortium with partners around the globe that have long-term experience work with climate-amplified epidemics and pathogens. And these partners include public health agencies. We are talking about the main public health agencies in the world, the Africa CDC, the WHO, and its associated agents, yeah, academic organizations, such as ours, public health institutes, and also funders and industry. We find very important that this program is also done with industry because they are the ones that are more likely to develop the diagnostic therapeutics and pandemics and vaccines. Yeah. So what we do, this consortium focuses in three main areas. Yeah. It focuses on the three areas to simplicity we name with ecology, understand ecological access, using very big data as we are doing now of climate change, of temperature, of precipitation, to try to really start tracking when and where epidemics are like to emerge. Advancing in epidemiology, we are talking about also big data analysis that involve mobility data from cell phone data, yeah, flight data, calculating the risk of importation of a pathogen, that when you link that together with the susceptibility, you can start guiding where to sample and when in space and time, and really our effect on evolutionary analysis of pathogens. So we have methods that can sequence any pathogen in the world, metagenomics methods, and target. And we have to look, because what we saw with SARS-CoV-2, pathogens can evolve to be more transmissible, to escape vaccines, and to escape diagnosis. So it's an arms race, and that's where we bring these three expertise together. Which programs we start? Yeah, we divide that program in different geographic regions in the world. We have a program in Climate Africa, Climate Americas, Climate Asia, Europe, and Oceania. I'm just going to give an example how three of the programs come together to try to identify and characterize epidemics in Africa. Despite having regional programs, we collaborate with them using the expertise. So, for example, in this initial program that I'm going to show you, we use a lot of the methods that have been developed in the Americas, especially Latin America, to trace arboviruses. This means epidemics caused by viruses and by mosquitoes, such as Zika, dengue, chikungunya, yellow fever. How to transfer that with Africa, and with American and European collaborators, how to use the big data to fast track that. Yeah. So, for example, the program has begun in Africa by mapping the analysis of arboviral data, producing genomic data, and training African laboratories to diagnose and sequence pathogens. Yeah. The first activity already has started, already had results. That's modeling and mathematical analysis of disease susceptibility and in Africa, and review all the activities that have been done. Yeah. Then we have offered and we are providing rapid genomic sequencing for any of dozens of viruses that are transmitted by different vectors, yeah, to any African country. So they can send the sample to us. We will sequence them for free, as we have done for 26 African countries during the COVID pandemic. But they can also request support where we give them protocols and many times send teams and diagnostics and reagents to be sequenced in the area. And then together to determine what we should be focused. We find that's very important that regions in the world and countries with local scientific divide what's to be done. Yeah. So this program built a very successful program that we had with African scientists. Yeah. During the pandemic, we led all the analysis of the COVID data in Africa country. We are talking about collaborating with hundreds of scientists. Our last scientific paper, our last results that come in Science, one of the top scientific journals, have 300 African collaborators. Yeah. And not only collaborators, but leading the process. Yeah. So that's what we are doing in Africa. We are basically answering a few big questions. Yeah. I'm still in the previous slide. Yeah. Yeah. A few big questions there. Yeah. When and where these epidemics are emerging. Yeah. What and how they emerge, what we know from previous data. Yeah. And then to analyze all this data to really develop where we should be doing. It's a program that happened. We meet every week with hundreds of African collaborators. As I speak here to you now, we have brought collaborators from 25 African countries to our lab to be trained on how to sequence these pathogens in the country. And we do that following what has been constructed during the pandemic, a very effective system of laboratories in Africa, which we being a specialized genomics facility of the Africa CDC and the WHO, we can offer this support for the African countries. And very important, we do that also with industry. Both Illumina provide reagents and with Abbott providing diagnostics. So we can make sure that we can identify these pathogens in the different African countries. Yeah. So in addition to that, what we do is our very long and very impactful program of capacity development. Yeah. So what we did, it is really transfer programs to very large groups, both to analyze data. For example, in August last year, we ran a very large program in South Africa, in Senegal, and in Panama, where we train people on how to do that. As we speak now this week, we have the large training program for 25 African countries. And in August this year, we are bringing to Africa the highest and most advanced virus evolution and molecular epidemiology workshop, or VIM workshop, really to make sure that we can not only generate, but analyze the data in the continent. Yeah. Just to summarize and to conclude, yeah. Over half of the non-pathogens in the world are expected to increase due to global warming. Yeah, Brazil and Latin America have large multiple concurrent arboviral epidemics such as dengue, chikungunya, Zika, and yellow fever. We have worked with them. We have sent many times deep in the grounds, yeah, to characterize these epidemics. We have learned a lot from that process. The predictions that Africa may be the next big place to be affected by these outbreaks, yeah. And it is of global interest that both this program that identify these new emergent virus and bacteria, Latin America, in Africa and Asia, is done as quick as possible, because this, as we know, climate change keep advancing, and the same epidemics will be hitting very soon Europe and North America become hotspots. So how we call this program is basic a health insurance for the world. If we can quickly identify the pathogens and characterize, we can really help the whole world that when that starts growing, you have the diagnostic, the therapeutics of the vaccines. And that's what we do based on the need to increase surveillance, early detect and respond to outbreak and epidemics. And that's why we find so important to partner with partners like Abbott for that process. So thank you very much. And here we officially launch the CLIMADE program.
Thanks, Tulio, for presenting CLIMADE. And did we lose the slide? Sorry about that. Okay, so I'd like to thank Tulio for sharing the great work you've done with your team, and we really are glad to be working with you as part of the coalition. So after seeing all of the potential impact that climate change can have on infectious diseases, there are things we can do today, especially you who are working in clinical labs. And so preparation really is critical to a quick response. That means that the lab can really truly play a critical role in protecting communities during outbreaks as they emerge. So that can look like a few different things. So first, we should be familiar with infectious diseases that are impacted by climate change. And we've seen some that Tulio has shared, and I'll go through some in a moment. Secondly, we can understand how your hospital system can be preparing today so that you can respond to new and existing viral outbreaks and epidemics. Because as we've seen, the patterns of infectious diseases will be changing as climate changes. And so in particular, there's a category of pathogens that are impacted by changes in precipitation. So storms and floods and water causing the pattern for infectious diseases like hepatitis A and influenza and the others listed here to become closer to people in ways that they haven't before. So precipitation changes have been shown to change the range of where vectors and animals who carry pathogens can live and thrive. And as climate changes, wildlife can come closer to people as they forage for limited resources and have different habitats. And as there's more water, there's also more wastewater overflow that can lead to contaminated water sources, and foodborne illness can thrive. On the other hand, climate change can also bring people closer to the pathogens. And that can happen when changes in precipitation have already changed how people are gathering. And as Tulio illustrated, that can also lead to migration of people in response to being displaced by floods and other water-related events caused by climate change. And so beyond the other factor, besides water changes, are also temperature changes. So that can be due to direct increases in temperatures and also fires and drought that can be secondary to those changes in temperature. And so there's a few pathogens that are on both lists, but this list is slightly different. We see things like dengue virus and Chagas and Crimean Congo hemorrhagic fever in this list. And so when these pathogens come closer to people because of changes in temperature, it looks like wildlife changing where they can live, and their habitats are changed when they have to move away due to climate change. And then warming can also allow mosquitoes to thrive. In particular, they can live in warmer temperatures more easily, and they can actually have higher rates of biting and have higher rates of viral replication in the vector itself. We also know that warmer temperatures bring people in closer contact with each other. And alarmingly, we also know that melting of ice in the permafrost can expose pathogens that we have not seen in a long time. In fact, this has been called Pandora's box. And so there's potentially ancient pathogens that could be re-released as we see melting waters due to higher temperatures. And so how does all of this impact what you can do today? Essentially, building infrastructure to screen out current epidemics can help prepare for the outbreaks that could be happening or new pathogens as a result of climate change. So that means that you can start working with ways to combat existing epidemics and pandemics that we're already dealing with today. Because if we're able to respond to those better, we'll be able to make a difference when new pathogens emerge. So things like opt-out testing to screen for multiple infectious diseases from a single patient interaction and sample can be a good step forward. These kinds of programs are going to be interacting with people who are already accessing the healthcare system, including people who could be difficult to reach otherwise. It also eliminates the social stigma of requesting a test. So if people are all getting tested, it doesn't seem like someone's singled out for just being tested for one of these viruses. This can also reduce the cost of expensive inpatient care if we're catching infections sooner because people are more broadly being tested. And then earlier diagnosis can also obviously lead to better engagement in care so that someone can get the care that they need earlier in their infection, which improves outcomes. So overall, building these processes in today can help protect the community in the future, not just with the current pandemics, but also to prepare for new pathogens that could be emerging. So some considerations for your particular lab could be how you manage your workflows. So this could be ensuring that you have an emergency response plan so that you're prepared if the unthinkable happens as a new pathogen enters your lab. You can also cross-train your staff so that they're ready and prepared to respond if needed. And having the flexibility to be able to use platforms and cross-train can also allow you to have people working wherever they're needed most. You can also make sure that you're prepared for changes in electricity in case there's an emergency, since that can be a critical aspect of dealing with an emergency related to climate change. You can also prepare for supply chain management. And that could mean that you're sure that your vendors can scale up to accommodate needs that might increase if there's an influx of cases that need testing for a particular pathogen that's impacted by climate change. And you can also adjust the way you manage your inventory looking ahead as you might see news coming about, perhaps from CLIMADE, about new pathogens that are likely to spread in your area. And then you can also perform surveillance of your laboratory test results. And that could mean that you're keeping an eye on what tests are being ordered and if there's a sudden spike in a particular pathogen that people are asking for test results on. And if those end up being negative, that could be an early alert system that there could be a new pathogen on the loose. And so with that, I'd like to thank you for listening to us today. And I would encourage any questions that you'd like to ask.
Well, thank you, Dr. Rodgers and Professor de Oliveira for that informative presentation. We will now start the live Q&A portion of our webinar. And if you have any questions you want to ask, please do so now. Just click on that Ask a Question box located on the far left of your screen, and we'll answer as many questions as we have time for. So let's go ahead and take a look and see how many questions. We've got quite a few coming in already. Our first question, what kind of information can we learn from viral sequences, and why are they important for clinical labs?
So I'll start, and I'm sure Tulio could add to this because we've both been working with a lot of sequences. But in particular, the sequences from viruses can be really important for clinical labs because as viruses mutate, we need to make sure that tests are continuing to detect them. And so if we're monitoring sequences for target regions of tests, we'll be able to ensure that tests still work or alert that they might not, and we can correct that. So sequences can tell us a lot that would impact diagnostic tests in clinical labs. I'm sure Tulio would like to add to that.
Yes, so I agree with Mary on the diagnosis, but can also tell you that sequence can also identify how a pathogen is evolving. So at the moment, our biggest fear as evolutionary biologists, I work with virus evolution, is that the current strain of H5N1 that's circulating avian across the world, that's decimating the avian species with over 100 million deaths that may evolve to become more pathogenic. So we want to have that information, especially if evolve to become human transmitted. We want to have that information before that happens. And that's where we should be sequencing this virus. That's a good example. Other way that's also quite important is related to resistance to therapies. For example, with HIV, we want to know if not only the diagnostic is effective, but if the therapy will be effective, because that would be the objective of a laboratory, to guide a lot of time clinicians on the effective therapy for HIV, for TB, for cholera. We want to know if the antibiotics will work or the vaccine. So there is many utilities for knowing that information for public health, for laboratory, and for patients.
Thank you so much. And our next question for Professor de Oliveira, this is actually a two-part question. Why do you think Culex does not receive as much attention as Aedes and Anopheles? And since Africa might be the upcoming hotspot for the next global pandemic, what are the chances of that pandemic being caught by Rift Valley fever, since it is a viral zoonosis with a large potential of outbreaks?
Okay, thank you. Thank for that question. That's a very precise question. So I do agree. I believe that, of course, we have been very focused on understanding the other two subspecies of mosquitoes, the Aedes and Anopheles. So that's the one that causes a lot of the outbreak of the other arbovirus. But Culex is the one that we worry. And we worry also for some of the virus adaptation from a species of mosquitoes getting adapted to the other one. Rift Valley has a pandemic potential. It's something that we are very aware. Any warning, so for example, last year there was a warning of a potential Rift Valley outbreak in South Africa. The warning, it came from basic data analyzing both mosquito concentrations, temperature, and water. We were quite prepared. We have sequence from that. That warning was a false warning. We didn't have any outbreak of Rift Valley in South Africa, but the year before we had one in Kenya, which we also have a team that collaborates there with our colleague at the ILRI, and we could classify and sequence the Rift Valley fever to see if it's evolving to be more transmissible. So that is a virus that is in our radar, and we know that as global warming increases, that's going to be more warnings of potential outbreaks of Rift Valley fever. And that's why we have to have a system to predict and detect very quick to make sure that before it's starting to become epidemic, that we have not only the diagnostic, but the therapeutics and vaccines for such a threat.
Thank you so much, Professor de Oliveira. Our next question from our audience, and I want to thank again our audience for these great questions coming in. How does this impact people living in North America or people outside Africa or the South?
Well, I would say I know, living in North America myself, that we have floods and hurricanes and a lot of natural disasters where these kinds of pathogens can thrive. And we're certainly not immune to the effects of climate change, with changes in pattern of the range where specific species of mosquitoes can live. So I don't really think this is limited to impacting the global South. I think this impacts everyone.
Yes, I completely agree. And especially if you just go a little bit back in time, very recently with the COVID pandemic, you will remember how unprepared was North America. It was Europe for that, despite multiple warnings. So we need to be prepared. So some of the pathogens, there is very direct implication. One thing that we learn when we detect the Omicron variant is that the virus and pathogens, they do not respect borders. They travel in planes, they travel in birds, they travel in containers of ships. So it's quite clear that large introductions could happen in America, and that could fuel outbreak. And in Europe, so that's why it's of interest of America, North America and Europe to invest in such program, because would it be much safe that you have detect these pathogens as they emerge, before they are being introduced and causing large outbreaks, At the moment, we know some of them that's already causing large outbreaks in America. For example, West Nile in Florida, Zika in Florida recently. We know that also with floods, you can have bacteria that the same way that you have cholera here, wouldn't be impossible to also have cholera outbreaks in America. So that's why we went to a global program, that could provide basic early warning and characterization. Really what we believe that would be a health insurance for the world there. And I really believe that both Europe and North America should invest in that program. So we already have big investors such as the Rockefeller Foundation, the Abbott, and other companies and funders that are seeing the importance of quick characterize virus and bacteria where they emerge before they become a global problem.
Well, thank you both very much. Our next question coming in, and we have time for a few more. What kind of data will be used to predict climate-amplified infectious diseases?
Okay, yes. So what kind of data it's used? We have two main sources of data. One is what we call ecological data, and that is, for example, data related to temperature, in a given location, data related to precipitation, rains, early warning of floods coming, of high cyclones. So this is data that we currently use very well to forecast climate events, So that's really important data to us to start linking to that. In addition to that, we have something that call sometimes intermediate hosts, the presence of a given mosquito or a tick or maybe a bacteria in the water. So we start linking this data of climate with presence of amplifying agents. And then you add the next big layer of data, that's data related to mobility, flight data, cargo data, where is likely to be introduced, what we call that the risk of importation. Animals such as bird flight routes. So imagine if you have an outbreak of pathogen X that can be transmitted in birds, and you have a direct flight route between that area to where you are, that should start ringing bells. And that's what the kind of thing that CLIMADE does. Most or big part of CLIMADE is not laboratory, it's data scientists that have access to very large computer clusters that can start linking and cross-checking data that can start highlighting where epidemics may happen. Two very simple cases, the very explosive epidemic of chikungunya associated with increase of 2 degrees last year, high rains and high mosquito population, the floods that had in Pakistan that fueled a cholera and dengue epidemic, the floods that happened in Malawi that fueled the largest cholera epidemic ever seen in Africa. So that's the kind of data that we want to be using real time to predict, and then we want to have the diagnostics and the way to respond in the right place before it happens.
Thank you so much. Now, are there recent examples of climate change impacting infectious diseases?
Yes, I can give like 50 examples, okay? But I don't want to overwhelm everyone. So we have, for example, resurgence, for example, when direct climate change, such as cholera, such as chikungunya, such as dengue, such as yellow fever, such as Zika, such as Rift Valley. So that's when you have higher number of warmer weather, more rain, and more vector. And we are talking about 6 pathogens that cause some of the large world outbreaks in the last few years, Latin America, Southeast Asia, and Africa. And then we also have the ones that when you disturb the environment, and we start destroying the environment, that animals get in much closer proximity with man. We talk about SARS-CoV-2. We are talking about influenza. We are talking about many different pathogens that would live naturally in the animals, but when the habitats are disturbed, they get much closer proximity with humans. You have the ones that are amplified by water. We have back cases of hepatitis E, with a terrible virus that we haven't seen outbreaks for decades. And that with very poor sanitation in some refugee camps or other areas, And we have cholera, you have E. coli, but that's normally amplified by climate events and high rains and floods. I hope I could give you a few examples. We are talking about respiratory, vector-borne, and waterborne pathogens here.
Thank you so much. Now, how does climate change impact something like HIV or hepatitis?
So I'll start with hepatitis because there's foodborne hepatitis viruses like hepatitis A or hepatitis E. And so whenever there's contamination of water sources with wastewater due to flooding, that's when you can start get contamination that makes people sick. But of course, hepatitis B, C, and HIV have a different method of transmission. And that can change when people are closer together and surrounded in new surroundings. And whenever you have migration of people, you have migration of those viruses too. So there's always that potential as climate change affects where people live that you can have changes in all of those viruses.
Absolutely. And it looks like we have time for just a couple more. How did climate change affect the spread of Japanese encephalitis in the Asian part of the world?
Yes, thank you. That's a very important question. For example, we know that Japanese encephalitis it's happened more common, for example, in Australia. Australia and the University of Sydney and the Australia Public Health Government is a big part of the CLIMADE program, and the leading scientist there is Professor Edward Holmes. So what they highlighted that again another mosquito outbreak that can be amplified. So Australia, unfortunately, may be one of the first developed countries that suffer for severe climate epidemics because they are in that area of the global south, and that's why they are investing also big all the top scientists, many of the top scientists in the country and the institutions in this program.
Thank you so much. And we have time for one more question. What surprises can be anticipated from the melting of the permafrost?
Well, there could be a lot of surprises there. I mean, these would be pathogens that have been extinct for thousands and thousands of years. And so unless we actually go digging in the permafrost to sequence them, it would be hard to know. So I don't know, Tulio, if you've seen any data like that before.
Yes, I think the one that scientists worry the most would be different pox and smallpox that could have been frozen for years and re-emerge. But another thing just to highlight is not always just something that exists and re-emerge, but as you start losing ice caps and forests, you have animals that would be living in more far away from the human populations coming closer. And a lot of times, many pathogens that we don't even know because we didn't have contact with the wildlife. And that's what I worry the most. It's not the ones that we know, but that's the ones that we do not know, such as it happened with the SARS-CoV-2 pandemic recently.
Well, Professor De Oliveira and Dr. Rodgers, thank you so much for your presentation today. Would you like to provide any final comments for our audience before we go?
My final comments is that if epidemics happen in one part of the world, they can easily happen in the other part of the world. So, and that's why we have to work as a global program. Yeah, this thing to believe that like that's a problem that will not affect me, my country, unfortunately, is not what we have seen with the COVID pandemics. Is not what we are seeing with many spread of pathogens to regions, geographic regions that we have not seen. And we are unfortunately entering, as we destroy the environment and climate change, on the age of epidemics. I know that no one want to hear about that after we finish a pandemic, yeah. But we also doesn't mean that we have to enter in the age of the pandemics, yeah? And that's why this kind of program to allow the highest level technology and support to detect pathogens where they may emerge, which we don't know where it is in the world, is so important, so that we can have the tools to control an epidemic before become a pandemic. Yeah, thank you.
Yeah, and I would add that we're in a position to be able to deal with these new pathogens. And so there are things that we can do to prepare so that we respond and avoid an epidemic from becoming a pandemic, like Tulio said. Because if we work together, we can actually apply those tools and make a difference. We don't have to let this get out of hand if we work together. And that's what I'd encourage anyone listening to do.
Well, I want to thank you both again for your time today and for such important research. We would also like to thank LabRoots and our sponsor Abbott for underwriting today's educational webcast. And before we go, I want to thank our audience for joining us today and for their interesting questions we did not have time for today, and those submitted during the on-demand period will be addressed by our speakers via the contact information you provided at the time of registration. And this webcast can be viewed on demand. LabRoots will alert you via email when it's available for replay. We encourage you to share that with your colleagues who may have missed today's live event. Until next time, take care, everyone. Be safe. Bye-bye.
Take care.